Here at Berkeley and also the Redwood Center for Science. And so, he has been, now everybody's really excited again about looking at your brain and saying, how can we understand some of perceptual problems and cognitive problems, but he's been doing this for a lot longer before it was cool, you might say. So anyway,
right. Thanks. Yeah. Right. Thanks very much. So I'll tell you today about a relatively new direction of work for our lab. And this work is kind of a synthesis bringing together ideas from machine learning and engineering and also neuroscience, an attempt to sort of formulate a new set of ideas that we think actually could contribute back to both of these fields. And so I should first begin by telling you about the people who did the work. And so this is the folks in the Redwood Center for Theoretical Neuroscience here at Berkeley and Evans Hall. And we're basically a group of faculty, postdoc students, many with backgrounds in physics, mathematics, engineering, and we're trying to bring ideas from these different fields to bear on the question of information processing in the brain. And so the folks whose work I'm going to be telling you about today are Alex Anderson, all of whom are here today, actually. So Alex is here, who developed this model of fixational eye movements and how we see with fixational eye movements. Brian Sheng and Eric Weiss, who developed this model of how you could learn the tiling of a foveated retina. And then also worked together with Pentti Kanerva, between Eric and Pentti Kanerva, who's also here, developing a model for how, basically how information could be integrated across saccades. Okay, so there's a lot of work now happening in engineering. You see and a lot of the work that people have been talking about here in this meeting that's inspired by neuroscience. And this is really exciting to us in the field of computational neuroscience that more of this is happening. And indeed, there's a lot to be inspired by. So even at the front end, just without even going into the brain yet, just looking at things in the front end, the eye is roughly equivalent to a 10 megapixel camera. And what's really remarkable about it is the fact that it's always on, at least 16 hours a day. And that's remarkable from an engineering perspective because people who build Google Glass know if the camera is always on, the battery drains in like 5 minutes or something, right? So just the process of imaging alone is very power hungry because of all the A to D conversion and compression that you have to do online. So this is an amazing feat in itself. And then you just look at the information going to the brain. The optic nerve here has about 1 million fibers in it or so. And you can basically come up with a pretty good quantitative estimate of the amount of information that's being streamed back to your brain. It's roughly about a gigabit per second that's inundating your brain, okay, 16 hours a day. And so this sort of begs the question of where does all this data go, right? It's not like there's some hard drive here in the center of the brain that's kind of collecting all this data. The data has to be assimilated on the fly. So as it comes in, your brain has to sort of figure out what parts of this data are new, what parts have I already seen before, what needs to be sort of assimilated and incorporated into my model of the world, and so forth. And if you didn't do that, then the data's gone, and you'll never get it back again. Okay, you don't get a second look. Okay, so all this has to be done on the fly. And it's really remarkable that all this is done to engineers, right, electrical engineers who are building circuits, that all this can be done in just 20 watts. Okay, so today I'm going to be talking about another really remarkable property of this front end, and that's the fact that the eye moves all the time. Okay, so there's no such thing as a static image to your brain. So the eye is constantly in motion, if not because of the eye movements within the head, but also your head is moving, your body is moving through the world. So the image data that's coming into the system is highly dynamic, and most of it is because of your own motions, because of self-generated motions, irrespective of the fact that the things in the world are moving as well. Okay, so I think this is really interesting because there's a place where we can go beyond inspiration. The biology, looking at how things are done in biology, sort of points out to us things that are important that we may have overlooked. And one reason why this is sort of a lot of times overlooked is because we're not actually that aware of it, right? Introspectively, we're not really aware of these movements. We're doing them all the time. This is just Yarbus's measurements of eye movements when you're looking at an image. For example, when you look at this woman's face, this is sort of a very prototypical pattern of how your eyes move over the scene. So these movements, they're not just kind of random scanning movements. They're very purposely generated movements. And you're really, for the most part, unaware that you're doing this. Okay, so there's not really kind of something that rises to our awareness as like maybe this is something we have to think about in building a vision system. But from the point of view of biology, it's immensely important. So if we look throughout the animal kingdom, we see just about all animals are moving their eyes in some way or another. Okay, so this is just one example of that, the jumping spider. This is a very old strategy in vision. So the jumping spiders go back to the Cambrian explosion about 500 million years ago. Okay, so these jumping spiders have been doing vision this way for a very long time. And so the jumping spider is remarkable because it has 8 eyes here. So 2 of these eyes here are very high resolution. They're lenses, very large lenses on the head attached to the exoskeleton. And at the back of these tubes, they form a very high resolution image. And then these eyes on the side have like fisheye lenses with a very wide field of view of the scene. And these eyes here, on this, sort of pointing forward to the side, have a not as wide a field of view of the scene, but and also have binocular overlap. So between all these eyes together, it has a full 360-degree coverage of the visual field. And so the jumping spider is interesting because, as compared to other spiders, it doesn't use a web to extend its sensory space. It relies on vision to hunt and to find prey. So it uses this low-resolution system on the side of its head to detect things that are moving, and then it orients its high-res system and scans it to identify objects. And it also uses its high-res system to identify conspecifics for mating. It does courtship dances. It can do navigation in 3 dimensions. It can solve 3-dimensional mazes, and so forth. So it's really a lot of amazing capabilities. Okay, so this is just showing a video of a baby jumping spider that's 1 day old. So this is like 1 day after it's hatched from the nest. And so what you can see in this video is these tubes move back and forth. Okay, and for these high-res eyes, you can see these things inside the head because when it's young, the exoskeleton is clear, so you can see through the head. And these eyes, those tubes are moving back and forth in this kind of scanning pattern, right? And so the reason why it's doing that is because the retina at the back of these eyes is not a 2-dimensional array. It's actually a 1-dimensional strip of photoreceptors. Okay, so that's what its retina looks like. So it gets a very high-res image formed here. And the sampling density is about as good as a cat. So it has about as good spatial resolution as a cat. So this is a predator. It hunts. It has these eyes that look forward. And its behavior actually is very similar to a cat. You can play with it. You can sort of take up your computer screen, move the mouse, and it will chase it, try to attack it. It will stalk it, right? There's this stalking kind of behavior and stuff and so forth. And so it takes this 1-dimensional array, and it scans it back and forth to build up an image. Okay, so there's a lot of lessons we can learn just by studying, just by reading about and looking at this animal, right? So one lesson is that it's not about the image, right? So in the computer vision, we spend a lot of time fixating about the image, and I do this too, right? We sort of think about, I'm going to extract pixels, features from the image. I'm going to do segmentation. I'm going to draw lines between the boundaries and so forth. I'm doing this all in the image. I'm studying the image. I'm saying, here's the boundary of Lena's hat and so forth. And we kind of get fixated about this. But when you think about, this system, like which image are you talking about, right? There's 8 images in there, being formed in the world. And, that's not an image we can really sort of think about doing segmentation on, right? So the strategy here is that the array of sensors here, these are just a set of measurements about the world, okay? And somehow this animal has to build up inside of its head a model of the outside world. Okay, there's nothing sort of overt about the outside world on the retina. It's not like an image we can just look at and see, oh yeah, that's the pattern that it's seeing. Okay, so it has to build that up inside its head. It has to somehow take the information from all these different eyes and fuse it together to build up this model. Okay, so it's about forming internal representations, not about the image itself. The second thing that's amazing here is that there's no learning, right? This animal is born out of the box, ready to go. Okay, so it's existence proof of what biology can do. Okay, this is basically what we're looking at here is behavior that's been optimized through evolution. And it's born with a very highly structured circuit. And I would argue a lot of mammals actually are also born with highly structured circuits that are ready to go. And then we need experience to fine tune it. Learning is obviously very important for us, but it's not like, sort of born with a pile of mush and we need learning to do everything, right? It's so that you're born with a highly structured circuit and then experience kind of fine tunes it. All right, so this really introduces 3 questions, which I'm going to be focusing about today. So one is this question of how do we see in the presence of fixational eye movements, which I'll define in a second. The second is, given that you're going to move your eye around the world like this, you're going to rely upon the strategy of constantly moving your eye, what's the optimal sampling lattice for the front end? Okay, so in all our CCD cameras that we build, the pixels are sampled in a highly regular rectangular array. Okay, but none of these animals do that, and why? And so can we understand that in terms of some kind of optimization strategy? And then finally, this question of how is information integrated across multiple saccades? So how you somehow build up in your head through all these different fixations, all these different views of the world. How do you build up sort of one unified model of the world? Okay, so let's address this first question of fixational eye movements. And I should say there's not much learning involved in this first question, but I think it teaches a very important lesson about how active vision works. Okay, so this is basically what it looks like inside your retina when you're looking at an E, a very small E projected onto the retina. Can you guys see this? I hope this, yeah, it's dark enough. So each of these dots here is a cone or a photoreceptor, a pixel inside your retina. And the subject here is staring, just asked to fixate. The fovea is a little bit off to the side here, so the E is just a little bit away from the fovea. And the subject is trying to hold their eye still. Okay, you're asked to hold your eye still, and this is what happens. This is what you're doing right now. So when you read the letters on the lowest row of a Snellen eye chart, their size is projected to about this big on your retina. This is how big they look on your retina. And they're moving around. They're constantly in motion. So this is the idea. There's no such thing as a static image to the retina. In fact, if you hold it, if you stabilize it, it will disappear. It fades away. Okay?
Just out of curiosity, how do you image this?
Yeah, how do you image this? So this is the work, amazing, miraculous work in Austin Reuter's lab, which is right in that building right over there in Minor Hall. And so it's a method called adaptive optics. It's basically adopted from astronomy. And so what they do is they use a fancy array of mirrors to correct for the aberrations of the lens. So normally with an ophthalmoscope, if you looked inside the eye, you could not see the cones. You could just see the blood vessels and so forth. But when you correct for the aberrations, then you can look inside the eye and see this kind of image in a live human retina. Yeah, it's amazing. And so, okay, so this is really neat. So there's a couple of yeah, this is Austin Reuter, right? Yeah, they're probably doing this experiment right now as we speak, right over there. Yeah, this is human retina. So it's really great because you can identify all the LMNS cones. And in Austin's lab, they can track this motion in real time. Okay, so they can track it while the subject is viewing like this, so they have complete control of the stimulus. They can hold the laser just on one cone and stimulate just that cone, and then they can ask the subject, okay, what do you see? They can move the laser to the next cone and say, okay, what do you see? Right? So you can really look at sort of perception at the pixel grain of your retina. So it really opens up this avenue for doing lots of really cool, amazing psychophysics. Okay, so there's really some amazing questions here. So one is, the fact when you do this, when you look at this, you don't see the motion, right? You're just looking at it, and the E appears stable to you. It's just rock solid. It's not moving at all. Okay, that's your perceptual experience. The second is that you can read the E, right? Despite the fact that it's always falling on different photoreceptors. Okay, so how is this possible? And so another question that arises naturally is like, is this a bug or a feature, right? Is this something that the system is doing on purpose, or is it just because the eye is kind of sloppy and the muscles can only sort of hold it so well? So I'll just take a vote here. So how many people think this is a bug? Nobody? Okay, so you all know where this is going, right? Okay, so I'm sorry, question?
Yeah, what exactly did you mean when you say it goes away if you hold the retina still?
Yeah, so you can so because we can track the motion, Austin can track the motion in real time. You can compensate for it. So you can hold this. You can take the stimulus and you can move it so that it totally moves with the retina, right? And now it's stabilized on the retina. And if you do that, it fades after about a second. Okay, so you can.
Like your perception of it?
Yeah, the world disappears. Okay, so if you could really paralyze your eyes, which people have done, some human subjects have done, they've taken paralytic drugs and then. You go blind. You can't see. So it's basically, there's an easy way to explain this is that the neurons that take in all this information have an AC response characteristics. So they basically filter out the DC component. Okay, so and the reason why they're doing that is because the image always is motion, right? So there's no use in coding DC because it doesn't exist. Okay. All right. So what they've shown psychophysically, in fact, is that this motion does help you. Okay, so this is recent psychophysics out of Austin's lab with Kavita Ratnam, a student in the lab. And so what they did here is to look at perception under three different conditions. One is the natural setting where that E is moving over the retina in its natural way. And another condition where it's stabilized. And so as I told you, if you move the E with the retina, it will eventually fade, but that's after about a second or so. And so they're looking at perception here under much shorter time frames, less than a second. Okay, so it's not due. So any differences are not due to fading. And then the other is where the motion is incongruent. So they can also introduce motion of their choosing. So they can make the E move in a way that's not related to the motion on the retina. Okay? And so this is basically the data. In 4 different subjects, you see that the blue bars are the natural, when you see the E with the natural motion of your retina, and the orange bars are when it's stabilized on your retina. Okay, so it helps you a little bit. And this is when the characters get really small, okay, when you're right near the Nyquist limit of the sampling array. Okay, so we can see here that this motion actually helps you. And moreover, it's not just the retina's own motion, but turns out any kind of motion actually helps you. So this is the dark blue bars here is where the motion is incongruent. And so, so basically any kind of motion helps you see better. Okay, gives you better perception. Okay, so now the question is how? How does the brain do that? How the brain now has to take all these spiking messages coming in from this highly unstable array and make sense of them and somehow build up an image of the world from it, or build up some kind of a model of what it thinks it's seeing. Okay, so the first thing we can show is that we know that you have to somehow average over time because the image is being relayed to the brain in the form of a spike train. And the only way you can get information out of these spikes is by integrating over time or integrating over space. So if you just integrate over time, that's not going to work. So if you just take each neuron's response here, so Alex made a nice little simulation of this. So if you just sort of consider what's going to happen when the E is moving around here, and now you make a realistic simulation of the spikes that are generated by that E as it moves around the retina. So this is basically like the spike trains that are coming in from different neurons into your visual cortex. Okay? This is the information your brain gets, and your brain has to somehow say, okay, there's an E there from these spikes coming in. Now, if you just integrate these spikes in place, so if each neuron just simply integrates this response over time, it would work if if the E was held still, but if it's moving, then it's just simply going to blur it out, and it basically destroys the pattern. Okay, so that's not going to work. Okay, so the problem we face here from a mathematical point of view can just be phrased this way: that you get a bunch of measurements. This is what you start with, right? You're given some measurements of the intensity at each point in space as a function of time, and what you want to infer is this pattern S. Okay, that's what you're trying to figure out. That's the E. S is the spatial pattern of the E. And what makes it hard is the fact that it's moving over time. Okay, so it's changing its position over time, and we don't know this motion. Okay, so we don't know the pattern, and we don't know how it's moving. And we have to estimate both of these things. And there's some corruption in the process. The spikes are noisy, the photoreceptors are noisy, and so forth. And actually, maybe I'm sorry, I forgot to mention one thing here. And that's, to ask the question, why might this help you, right? So why might this help you? Let's just go back to this picture here. So the one reason why we think it might be actually a good thing for the system to do is because all these different photoreceptors here are different types. Okay, so some of these photoreceptors here are L cones, some are M cones. As you can see here, they're not stacked in a totally regular array. There's gaps between the cones. So if the E lands in between the gaps, it's not going to be seen as well. If it lands on an L cone, it's going to be seen differently than if it's an M cone. These cones probably have different gains. They probably have different offsets. And so you don't want to be sort of dependent on the idiosyncrasies of where you happen to land on the retina, that the letter is going to look different, right? And so one way to solve that is by averaging its appearance over different places in the retina. Okay, so I just wanted to get that in. So that's sort of where we're going here. All right, so here's the problem we have to solve. And okay, so the way we can sort of think of this is that we want to figure out what the pattern is and what the motion is. Now, if we knew the motion, if we knew how the eye was moving, if somebody told us that, then the problem would be pretty easy. We could simply take the image and compensate it by take the image data, compensate it by that motion, and then integrate over time. And that would do a pretty good job recovering the pattern. But of course, the problem is we don't know this motion. So we have to estimate the motion as well. How would you estimate the motion? Well, one way to do that is if we knew the pattern, right? If we knew the pattern there, then we could ask, well, what's the shift that best explains the image data at any given point in time, right? And that's one way of estimating the motion. But of course, the problem is we don't know the pattern, right? So this is a chicken-egg problem. We have to estimate these two things at the same time. Okay, so we need the motion to estimate the pattern, and we need the pattern to estimate the motion. Okay. So the argument is that you can't. It's not like we can build one box which estimates the motion and another box which estimates the pattern. These two things are intimately intertwined, and they have to talk to each other in order to figure it out. Okay, so this is the model that Alex developed, and it's basically based on just building a probabilistic graphical model to infer these things out of the spike train. I should mention that we're basically riding on the heels of some really nice work that was done prior to us by Yoram Burak in Marcus Meister's lab. But in their model, they had to make a number of restrictive assumptions about the nature of the pattern you're recovering, about discrete motion and so forth. And so we're trying to loosen those assumptions and build a more sort of biologically realistic by a physically realistic model of how this might happen. Okay, so here's the idea, is that you start with, this is the observations you're given are these spikes coming in from the retina. And from this, you want to infer the pattern that's present there and how it's moving. Okay? And so if you have a model for how these spikes are generated from the pattern and from the motion, then you can just kind of work backwards using Bayes' rule to figure it out. Okay, so this is just the problem we want to solve, is if we can write down the probability of observing a set of spike trains given a certain pattern that's present there, and we just want to find the pattern that best explains these observations coming from the retina, okay, these measurements. Okay, so in order to do that, as I said, we have to have some estimate of how the position is moving around at the same time. So we have to estimate both of these things simultaneously. So in order to estimate the pattern, we have to estimate the motion. And what Alex showed is basically you can just approach this by building an EM algorithm that iteratively sort of goes back and forth and tries to estimate these two things. So given the current estimate of position, update the pattern, and given the current estimate of the pattern, update x, and you just go back and forth between these two things. Okay? In a loop. And really, it's just happening simultaneously in the algorithm. Okay? This is just like two populations of neurons that are settling simultaneously to a solution. And so they derive a set of equations just by basically doing gradient descent on that objective of trying to find the most probable pattern and set of motions that explains the stimulus. And you get these set of update rules, and they're probably better explained in terms of a neural computation that you would do. So the kind of neural computation that we end up having to do for that is shown here. So again, we're proposing then, the hypothesis here is in order to solve this problem, the brain, somewhere like in the primary visual cortex there, in the input layer, we basically need two populations of neurons. One population of neurons which is estimating the pattern, and another population of neurons that is estimating where the eye currently is looking. Okay, so this is like a sort of a map, a position estimate of where you're currently looking, and this is an estimate of the pattern being built up. And here's the spikes coming in from the retina. And so the basic idea of those equations is saying, well, in this population of neurons that's representing the pattern, those neurons are multiplicatively gating these connections from the retina to these neurons in the cortex. Okay, so where the information goes, where it's routed into the cortex, changes as a function of time depending on your internal estimate of the position. Yeah?
Is this a soft return of the switcher circuit?
It's a shifter circuit. Yes, it is a more neurobiologically friendly version of the shifter circuit, I would say. Yeah, derived from Bayes' rule, right? So what Jan LeCun is referring to is an early paper by Charlie Anderson and David Van Essen. They wrote back in 1987 where they proposed this idea that the cortex must somehow be dynamically shifting the input signals to do this compensation. That was inspired by some findings from Gian Poggio's lab with Brad Motter, showing basically that somehow these representations in V1 are stabilized in the fovea in response to these small fixational eye movements. So there is some evidence for this, that you build up stabilized patterns, stabilized representations in the cortex. Okay, so that's one thing that has to happen. And then the second thing for estimating this position map, the idea is that you're going to propagate that forward in time, much like in a Kalman filter or in a hidden Markov model. Okay, so you start off with some estimate at time t of where you think the pattern is. And then you propagate that forward one step, so it gets a little blurrier because there's some uncertainty. So we're basically using here just a simple diffusion process to model how things are moving over time. One could certainly improve that with a more realistic model of the eye motion. Then you combine that together by using your pattern estimate. So this is your current hypothesis of what you think the pattern is. It might be sort of initially fuzzy estimate, and cross-correlating that with the spikes coming in to get sort of what's the best estimate of, based on the pattern and the spikes, what do I think is going on? And then by combining these two estimates or these two distributions, we can now get an updated uncertainty, and this just propagates forward in time. Okay, so these are the two computations that are going on in parallel. And all the computations that are being done here are basically sort of, they're all computations that could plausibly be done in a neural circuit. There's no reason why, nothing we know why, V1 could not be doing these kinds of computations. So here's a very simple simulation of the model. The idea, yeah.
Are those literally, the R's are binary, it's not real value? Right?
That's right, they're binary spikes. Yeah. Okay, so here's a simulation of what goes on. So this is that Alex made of, here's sort of a little sort of kind of a regular hexagonal sampling array in the retina, and the E is moving around here due to drift movements, and this is a simulation of the spikes coming in from on cells and off cells in the retina. And this is just showing then the idea of how the pattern gets built up over time. Okay, so just run that one more time. So it starts out with a kind of fuzzy estimate and that estimate gets refined over time. So the motion helps you build up this estimate of the pattern. At the same time, it has to estimate the motion, and this estimate of the motion is done from its estimate of the pattern. Yeah?
Does the brain get no feedback about the movement itself?
Yeah, good question. So this kind of subject to debate. There is some evidence that there may be signals about this drift. There's certainly signals about the microsaccades, but the motion here we're mostly looking at is the drift, which is smooth. And whether or not there's neural correlates of that the brain is getting is kind of unclear. But Austin's experiments seem to indicate, the ones I just told you about, that if you introduce motion that's not correlated to the eye zone motion, it still helps you, right? So that seems to indicate that the brain is not relying upon these external signals about the eye's motion. It somehow has to estimate whatever the motion is there to do this.
Bruno? So,
Yeah.
so in this particular model, it reconstructs E better than a static E?
Yes. Yes, that's being shown here. So here's just comparing the performance. If you took the E and just held it there in place, this is the no motion case in blue. If you just sort of integrate it over time, okay, you gain from integrating over time, but not as well as if you move it. Okay, so if you move it, it's going to help you. Okay. And that's because you're basically getting the benefit of moving this E over, in and out of the gaps over different photoreceptors. And we haven't actually in the simulation put in the different gain factors and so forth that could be here. Okay, so this is bound to improve if we put in more of the heterogeneity that actually exists in the retina to build up an image. Yeah.
Could there be an extra loop that moves the eye so that this, in other words, there are obviously muscles that are doing this saccade?
Yes. Oh, there's these fixational eye movements. They're sort of composed of two parts. One is the smooth drift.
Right.
And whether or not that's just like the muscles relaxing versus actually being driven in a continuous way by the muscles, by neurons, is I don't think entirely clear. But then the microsaccades.
Could be another optimization loop.
Yeah. Oh, I see. That's actually kind of like purposely trying to drift in a certain way. Yeah. Good question. We're not considering that. We're just sort of taking the drift as given. That's right. Okay, so another question is, what kind of pattern you're building up. So in that model I just told you, you're kind of just building up a pixel representation. But we kind of, we don't really think that the brain is in the visual cortex is literally building up like a pixelated image of what's in the retina. It builds up this representation in some kind of a feature space. And so the way we've approached that now is incorporate a dictionary. So this is incorporating a prior over the pattern. We're saying, well, whatever pattern you infer here, I'm not going to let you infer just like any old pixel pattern, like some noise, whatever. I'm going to say, well, it has to be some kind of a structured pattern. So one way of forcing that is to say, well, I have some dictionary that this pattern is described in. We'll learn this dictionary according to the statistics of the images coming in, and impose a sparse prior on the coefficients. Okay, so this is a way of imposing a structured prior on the pattern that we infer. So it's just like a sparse coding model. So now the idea is that the pattern we infer is actually being represented in terms of these sparse coefficients, which would look like sort of like oriented receptive field neurons like we see in V1. Okay, and this is just an example now extending this to going beyond E, going to the world of MNIST, which you're going to see a lot in this talk. So just now, for looking at MNIST digits, we can learn a dictionary for MNIST in terms of little oriented features. And now take an arbitrary pattern like a 2 and move it around, or a 3, or any kind of MNIST digit, and now we'll build up a nice representation of that pattern. Okay, and now we can show that here, incorporating this prior, this additional constraint over the shape, having prior knowledge of the sort of space of shapes that we're likely to see there, actually helps you. Okay, so this is just showing that with a sparse prior, you do better than with no prior. Okay, so I think this is important to think about because it's really like a sort of a microcosm of this bigger question of these sort of big saccades that we make all the time. Okay, and the first point is that even when you're holding your eyes still, the brain doesn't want you to hold your eyes still, right? There's some advantage to moving the image, right? It helps you, and we sort of have a good sort of mathematical engineering account for why that could be, and a hypothesis for the kinds of computations that V1 might be doing that support that. Okay, so let's go on this other question, like the main saccades that we make, these big sort of eye movements traversing around the scene. And to do this, I'm going to start by putting you in the role of a deep network solving the ImageNet challenge. Okay? So I give you this image, and what's the right answer? Dog. Wrong. What's that? Oh. Porcupine? No. Okay. You guys are not very good at ImageNet, right? Okay. You're horrible. So the right answer here is Pomeranian, right? So if you said dog, you're wrong. If you said dog sitting on a bed, you're wrong. If you said dog holding a ball sitting on a bed, you're wrong. If you said, blah, 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 I mean, anything, that's wrong. So it's kind of like the point here is that a scene is not just one thing. It's a composition of things, right? So I'll give you another example. So this scene here, what's the right answer? Okay, well, you guys are horrible again. All right, okay, so the right answer is Afghan hound, all right? And so if you said two people and their dog, you're wrong. Two people, a dog in the mountains in the background, you're wrong, okay? So the point I'm making here is that, when we sort of give these neural networks a task, to sort of take a whole image, a whole scene, and label it in terms of a single word, it's a very artificial or actually ill-posed problem. Because you're sort of taking this whole problem of perception and putting it through the linguistic bottleneck. And there's sort of very little, the bandwidth of what we can get out of our mouth is very limited. But the percept of what lives in our head could be humongous, right? And as Fei-Fei Li likes to say, you could write a novel about what you see in these images sometimes, right? So the level of depth you could go to in sort of describing what's here is enormous, okay? But the first point here is that a scene is really a composition of things and spatial relationships among those things. And the second is that when humans view a scene like this, we don't take in the scene all at once. We use attentional strategies to decompose it and analyze it one piece at a time. So attention is a serial process that moves over the scene either by eye movements or by an internal covert window of attention that moves over the scene and sort of binds these things, binds components of the scene together one at a time. So we do this in a kind of serial fashion. And there's loads of evidence from psychology about how we analyze scenes like this. This is the subject of visual attention. And so recently there's been some really nice work in the neural networks community building recurrent neural network models of attention, which we found very inspiring. So Brian basically took inspiration from this work. And so this is work, there's a number of different models like this came out of DeepMind. This is one particular model called the DRAW model from Carol Greger, who was actually used to be a student with Jan. And so the idea here is, again, rather than just sort of take in the scene, you give it a sort of a scene with an MNIST digit in one part of the scene with a bunch of distractors around it, and rather sort of take in the whole scene and sort of trying to answer it all at once, it has this glimpse window which can move around the scene. In this case, it can also rescale the glimpse window around the scene, but the neural network is basically trained how to move this window around the scene, initially starting from some kind of big default view, how to move its glimpse window around the scene so it can answer the question and say, okay, there's a 0 in this scene. Okay, so just analyzing it one part at a time. So the key idea is introducing this idea of a glimpse window and a neural network that controls it. And so the question we wanted to ask here is, okay, well, given you're going to move this glimpse window around the scene, how do you want to sample the information within the glimpse window? Okay, so in their model, they had a sampling lattice within the glimpse window, just like a fixed number of sample nodes and a rectangular array, and that's simply rescaled and repositioned to wherever the window goes. Okay? But one thing we know from just studying eyes of animals is that they're not built that way. Okay, so almost all animals have a spatially inhomogeneous sampling lattice, and this is especially pronounced in primates, where you have a high-resolution region in the fovea, in the center vision, and a very low-resolution region in the periphery. And that's not due to the packing of the photoreceptors, that's due to the packing of the retinal ganglion cells, which is the set of neurons that sample the image and whose axons form the optic nerve here exiting the eye. So the sampling by retinal ganglion cells is very dense in this region, and it's very sparse in this region. And it falls off gracefully in between. Okay? So this is the kind of spatial sampling lattice we have in our retina. And this is another view of it from a chapter by Anderson Van Essen. So the idea here, just each dot here depicts a retinal ganglion cell. So the packing of the retinal ganglion cells is very dense in the center, and it falls off with eccentricity. So the way people think about this in the field is kind of in terms of intuitive kind of thinking about this, is that the reason why you have this solution is there's a trade-off between having high resolution and a wide field of view. Okay, so it gives us both of these simultaneously. So if you were going to take the same number of resources that you have here and pack them all uniformly with the same density you have in the same center. So if you said, I want to have that density everywhere, and I'm unlimited to having this many resources, you'd basically be able to subtend about 6 degrees of visual space. Okay, so it's like looking through a little like peephole at the world. Okay, so you would have excellent resolution, but you'd have tunnel vision. And you'd have to sort of hunt and peck around to know how to see. On the other hand, the other extreme, if you took the same amount of resources and you spread them out uniformly over the entire 150 degrees squared field of view, then you would have wide field of view, but you would have low resolution, just extremely poor resolution everywhere. In fact, your resolution would be so poor that even the largest letter on the Snellen eye chart would look like a blob to you. Okay, so you would not even be able to resolve the largest letter on the Snellen eye chart. So you wouldn't be very good. Yeah.
Sorry, quick question. So why would you have sort of like tunnel vision if you kind of at the same density but all over the?
But you're saying well, it's not all over. You're saying I want to have the same density. And so if I'm limited to the same number of resources, I'm not going to let you have any more neurons. Take the same number of neurons you have here, right, and pack them at that density, then it would only take you out to about 6 degrees of diameter. Okay, that's it. You're done. That's as good as you can do. Okay, so that's kind of the intuitive argument. But now that makes sense, right? That's perfectly reasonable. But can we show that quantitatively, that this emerges somehow as the optimal sampling strategy? So this is the work that Brian Eric did by then exploring this in the context of the draw network. Okay, so this is just the draw network here out of DeepMind. And so here's the idea of the glimpse window, right? You have the scene here, which has some stuff in it. And here's your glimpse window. Okay, so the glimpse window has some samples in it, and these samples within the glimpse window are just fed to this array here. So this layer here just simply represents, one for one the contents of the glimpse window of these neurons here. Okay, and then that representation is fed to a recurrent network, which feeds back on itself over time. And out of that recurrent neural network, we have two sets of units, one which represents the object identity, so what's there in the scene, and another set of units which represents where to look next. Okay, and the simplest system is trained end to end to output the identity of the correct digit. So if you give it a scene like this, it should output 1. If you give it a scene like this, it should output 9 after it's looked around a few times. Okay, so it learns how to, not only to recognize the digit, but it learns how to move its glimpse window around in the scene, which is very interesting, I think, from sort of a neuroscience or psychology perspective. Here we have a neural network which has learned how to control its attention. Well, what does it learn to do? Maybe we could learn something from that. Okay, so the question we're going to ask here is now, once we can train to do all these things, but we're going to add now another degree of freedom that we can learn, which is how these sample nodes should be allocated within that window. Okay, so we can start them out uniform, but we can let them evolve through learning. So we could just make those additional parameters the position of each sample node and the radius of its receptive field. Okay, those are just 2 additional parameters per sample node we can add, and now we're just going to train those by gradient descent, the same as all the other parameters in the network. And this is what happens. Is that you initialize your glimpse window to an array that looks like this, and now you let it start learning, and the glimpse window starts to move away from that solution, and it moves towards the solution where it chooses to take a number of its sample nodes and move them out to the periphery with large receptive fields, and take another set and pack them into the middle with higher resolution. Okay? So stochastic gradient descent has told us that this is a good way to build the front end of your visual system, right? With no constraints, no additional constraints. We're starting off with that and it says, no, I don't want to be that, I want to be that. Okay, so this is just a nifty movie of this taking place. Oops, I'm sorry. There you go. Okay, this is just a movie of that. This is the process through learning, so many iterations, many epochs of learning, watching that solution evolve. Yeah?
Is that actually shrinking the size of the units in the center, or is that just to
Yep.
show it?
No, it's shrinking the size of them as well, absolutely. The size of that dot shows this learned size of the receptive field. So that was a learned parameter. So it's basically shrinking. In the center, they're shrinking the receptive fields, and out here, they're expanding the receptive fields so they can better sample space. Okay? And this kind of makes sense, a reasonable solution, right? Because, again, we want to have high res to do pattern recognition. We want to have high res to recognize the digit. But if we had tunnel vision, then we're going to be doing hunt and peck. to figure out where to look. So I'm going to put my feelers out here. I want some feelers in the periphery to sort of tell me where, well, where I should go next, right? And so that's what we need things in the periphery for. Yeah.
How do you put a conv filter on this if they're moving?
This is not a convnet. Is that what? Is that what you're asking?
Yeah.
Oh, yeah. It's not a convnet. Right. Spatially homogeneous. Okay. So so here's another interesting thing about it. The solution that emerges is, so here's showing what we get, two different solutions for two different data sets. So in data set 1, the digits all have the same size. In data set 2, the digits have different sizes, so variable size digits. And so you can see sort of a smoother, kind of more gradient looking solution emerging when they have different sizes. But now what we can do, this is the kind of cool thing about a simulation like this, is that we can entertain the possibility of having an organism that has a zoom lens. Okay, so there is no such animal that we know of that has a zoom lens, because all animals that move their eyes, they simply move their eyes. They can translate their retina, but maybe in another planet, you might find an organism that has a zoom lens and it can kind of zoom in and out on the scene. And so if you do give it a zoom lens, so now the system, like the original draw paper, it can change the size of its glimpse window as well. And now let the sampling lattice evolve, it chooses not to build a fovea. So the solution, it does not emerge in this condition. And that kind of makes sense, because if you can zoom, then you don't have to sort of rely upon this this kind of foveated system, right? You can just use your zoom lens to control the scale. So anyways, okay, I think that's neat. It's a little prediction that would come, maybe an experiment we could never do to evolve such an animal, but yeah.
So zoom lens is why then no animal with a zoom lens?
Why is there? Don't ask me. But yeah, I mean, yeah, it's a good question. I'll just speculate. I mean, mechanically, it's going to be a hard thing to do, right? You'd have to sort of figure out a way to change the distance of the lens, sort of deform the eye in some way. And mechanically, maybe it's just too hard. I don't know.
But isn't our attention mechanism in the eye, the saccade, kind of like a zoom lens?
The internal, there's evidence for like an internal zoom lens in the brain. So when you look at a scene like this at any given time, you can show that there's a certain scale that you're attending to. I'm attending your face right now, or I might attend wide, right? But that's all internal.
The zoom here is a condition on the diameter of the senses?
No, this is on the diameter of the whole glimpse window. So in this model here, the glimpse window can only move in the scene, just translate only. So it can change its position, but that's the only thing it can do. So in this model here, it's like the original draw model. The glimpse window can both move, and it can also change its size, right? And as the glimpse window zooms out, then all the same neural sources are spread out, right? It takes all these dots and will just simply spread them out, right? And if the zoom lens goes down, then all these would just simply scrunch together and go down, yeah.
Can you also speak at all to how the glimpse window moves around when it tries to find the number in the scene?
Like, is it just kind of, yeah. So I'm going to, Ryan doesn't have to answer that.
The test is pretty easy. Usually it just finds a digit pretty quickly and just zooms in and makes little adjustments to sort of classify it exactly as possible.
So you imagine that for those spiders, which have basically several different eyes with different zooms, they would not need sort of this kind of spread out, because they would use.
They get this foveated like eye with different cameras. They have, with different eyes. One eye does the center part, and the other eye does the kind of the big periphery part. That's right.
So maybe an argument for why there aren't animals with zoom lens would be that if you did zoom, then you can't pay attention to all the stuff that you're not zoomed in on, right? You still want to be aware of, a line.
Yeah, that's right. That's,
So the software zoom is
yeah, right. Yeah, okay, good. We're doing armchair, evolutionary biology, I guess. Yeah.
So what about those animals that have phoveas of different shapes, like rabbits have a horizontal?
Yes. Yeah, that's great. So rabbits have these strips, and so many sort of animals that are low to the ground. Their sampling lattice is, kind of organized around that structure. Birds, some birds have two foveas. So I think one cool thing about this framework, now that we have these recurrent neural networks that do attention, maybe we can take these systems and immerse them in different environments. So you simulate an animal that's low to the ground that has to hunt in that kind of world, or a bird that's kind of, give it different tasks, and see if different kinds of foveas emerge in these different task regimes. Opens up a lot of possibilities. I think that'd be neat. Yeah. Okay, so then we could compare the result. This is just kind of early days. It's MNIST world, so I have no right to be sort of comparing this to biology yet. But we can try. And already it's pretty promising. Is that if you just look at the sampling interval, we can just look at the sampling interval as a function of eccentricity. So the spacing between adjacent samples as a function of eccentricity that's learned in the model. You get this sort of linear increase with eccentricity, which is very similar to what you find in the biology and how the ganglion cells increase their spacing with eccentricity, and also in the diameter. So if you look at the diameter of the receptive fields, it's also increasing. It sort of saturates out here, but it's pretty close. So one thing we want to do now is actually sort of take this model and crank it up to more realistic settings, richer, bigger scenes, to see how these results change. Okay, but I think one lesson we've learned here is that there's a sort of intimate connection here between this foveated sampling strategy of the retina and how the imaging system is used in this active vision context. So it doesn't really make any sense to build a visual system this way if you're just going to hold the camera still. Okay, so there's a lot of attempts in technology. So there's these really interesting efforts out there to build foveated cameras. So actually where the sampling array is like that, which is really wonderful and cool. But it would only make sense to use this kind of a system if you're going to move it around in an active way to search a scene. And we think we have now a model that can show us how to effectively take the information that comes in from such a foveated system and utilize it effectively to do a search task. Okay, so the next question is how information is integrated across multiple fixations. And this is a really interesting question from the point of visual neuroscience, because it's another place where we have a complete disconnect between our sort of introspection or perception of the world and what we know about what's going on inside the brain. So here's the remarkable thing, right? I just told you you're moving around all the time, okay? So that means the image data that's coming into the primary visual cortex is changing 3 to 5 times a second, dramatically. It's being completely replaced with a different image, okay? So the representations in V1, V2, V4, all these cortical areas back here in the back of your head, every time you've removed your eye, everything we know from the physiology says they're updated with a completely new image of the world. Okay? Yet, it appears to you like the world is stable, right? And all of these sort of different fixations, you're largely unaware of them, and it just seems like one big world that you see in high resolution all the time. And it's stable. It's not moving around, right? And you have no sort of sense that these are coming into like little pastiche, little patches about the scene, right? They're somehow seamlessly stitched together, and they build this kind of coherent perception of a stable world, despite the fact that the input is highly unstable. Okay, so one question is, well, where does that exist in the brain? Do we find an area of the brain where it looks like the worldview part of the brain, where neurons are kind of building up an internal representation of the world? And the answer is no. So far, no one has found any kind of neural correlate of some area that does this directly. Okay, so we find neurons that are modulated by eye position in various ways, but there's no clear explanation for this. Neuroscience has zero explanation for how this happens. So in the meantime, we can try as modelers to do this, right? So we can try to build neural models. What would you do if you had information coming in this way, and we're trying to use information from these different fixations to build up some kind of an internal world model of what's out there in the scene? How would you do it? Okay, and just maybe think about that. Okay, so that's what we did here, and so here's our initial thinking about it, and basically the solution is pretty simple. So I'm going to argue there's basically 2 things we want to think about as you're sort of fixating around. We have the position of the glimpse window, or the position of the eye at any given time, that we need to think about encoding that and keeping it around somehow, and the contents of what I saw at that location at any given point in time. What was there in the fovea, or what was I looking at? And somehow we need to bind these 2 things together. Okay, we need to bind these 2 things together because I'm about to move on to a new location. Okay, so as I move on to the new location, I don't want everything that I saw there to be forgotten. Right, I need to kind of build it up. So I have to somehow take these 2 things, bind them together, and keep it around. And so the idea here, the simple idea we're going to propose, is that if we have a way of binding these 2 things together, then we could represent a scene simply as a superposition of these bindings. So I take, what I saw there, bound together with where I was looking, plus what I saw there, bound together with where I was looking, et cetera, move around the scene. And if I just add those together, that is the scene representation. Okay? That's a sort of one way of giving kind of a holistic representation of the scene that's built up. So now one question is, how do I do binding? Right? I'm saying, well, you bind these two together. What does that mean? Because this basically goes back to the question of variable binding, right? Which Christos has been addressing as his model, and many other people have kind of been looking at this problem. And this is something that's been argued for a long time, whether or not you can do variable binding in neural networks. Gary Marcus has argued that it's a crucial property of cognition. Unless you have the ability to do variable binding, that you sort of can't explain many of aspects of cognition. But there's been kind of a sort of nascent growing movement over the past 20 years or so of work that showing basically how you can do binding in neural networks in connectionist-like structure. This sort of originated from the work of Tony Plate, who was a student with Geoff Hinton in the late 1980s. And so he published his thesis as a book here, and it's a sort of a method what he refers to as holographic reduced representations. And basically, one way of thinking about it, if you're familiar with his prior work of Smolensky, so Smolensky had a solution to how to do these bindings using the outer product. And the problem with the outer product rule is that it explodes the dimensionality of the space. Okay, so it doesn't sort of scale very well. And so we think what Plate basically showed is there's a way to do this, to the binding, without exploding the dimensionality of the space. You can keep the dimensionality the same. Okay, so you have a population of n neurons, and I can always stay with n neurons, and I can do this. And it's a fully distributed representation. And what Penty has been showing now, and building on this in what he calls his framework of hyperdimensional computing, is there's a set of operators over this space. So we basically work with very high dimensional vectors, usually between 1,000 to 10,000 dimensional vectors. And you use multiplication for binding, so simply element-wise multiplication of these vectors is the way you do binding. Addition for combining things, and there's a set of operators and inverses on this space. So I'm going to sort of illustrate this briefly in this example, but I highly encourage you to read chapter 6 of this book and this paper here. They were truly an awakening for me, and I think there's a lot of potential for what these can do if we incorporate these ideas into deep learning architectures.
Which one are you recommending, Kanerva or
both?
Both.
Yeah, absolutely, required. Yeah. So this is the origin. I mean, Penty, I know what you add to that, but this is kind of the origins. I mean, it was Tony Plate's work, and then Penty is kind of building on this. Are there people who've been contributing this, like Ross Gaylor, and others you would recommend?
Oh, I think Chris Eliasmith's work.
Chris Eliasmith's work on what he calls the semantic pointer architecture is also based on this idea. Yeah. Okay. So here's the basic idea of how we're going to employ those ideas here. So this is work that Eric Weiss has been doing. Is you have a scene like this, and now I'm going to take this glimpse window and move it around the scene. I should mention, by the way, that draw network that we just talked about is basically doing this somehow, right? The draw network has this recurrent neural network that's taking in information from all these glimpses and allows you to build up a representation over some time somehow that can answer the question. And the way we sort of got started on this track is that Eric was kind of interested in, well, how is that happening? What's the recurrent neural network doing? How can I sort of understand the representation that's being built up there? And so this gives us a way of understanding that, what's happening. Okay, so the glimpse window is currently here at this location, and what we're proposing is then say we're going to take the location where we currently are, it's really just two numbers, and we're going to code that as a high dimensional vector. So in this case, about 1,000 elements. Okay, so we're going to learn some function. We're going to learn this function f that maps the two-dimensional position into this high dimensional vector r. And that encodes the current position of where I'm looking. So you might imagine this is something the eye movement system is doing. I have access to this information, the eye movements, and that's encoded into this high dimensional representation. And now this is the contents. This is sort of whatever I'm getting from my sampling array of my glimpse window. I'm going to take that content, what's in my glimpse window, and I'm going to learn a nonlinear function that maps it into a high dimensional vector v. Okay, we're going to learn this function here. So we're going to have a vector here of the same dimension as that for representing the contents of the glimpse window. And now how do we bind those two together? We bind them by element-wise multiplication. So I'm just going to take this times that, and now that is those two things bound together. And I'm going to add that into my short-term memory. So M is basically a short-term memory that's being built up about what's out there in the scene. So at the first glimpse, let's say the M is empty, it's all zeros, and this first glimpse is these things are bound together and added in there. And then the next glimpse, I move on, and those are added in, and so forth. I just accumulate basically accumulate these bindings together. Yeah.
So you are multiplying these two vectors element-wise, so they have to be the same dimension.
That's right, that's right. They're constrained to be the same dimensionality.
But isn't it sort of the encoding very different information?
Yes. Is there a reason why they should be this? Well, it's basically, I think the short answer is because that's the way it works. Right? I don't know if you have a better way to answer. I mean, we're just going to, yeah, so it's a, yeah.
It needs to be because we want, the thing we're after this is like a unified representation space for all possible information, right? So, I mean, and this is, this is vision, but I think I think one of the motivating ideas behind this approach is hyperdimensional computing in general is to try to address like how would we represent like a whole bunch of modalities or pieces of information in one representation.
One workspace where you could bring disparate things together and into that workspace and bind them together and now manipulate those representations.
Somehow the dimension is high enough.
It's high enough, that's right. You pick a number. If you're building it, how biology does this and how we map it onto biology is sort of another question. But you pick a number, in this case 1,000, and you go, okay? Yeah.
When you have high enough dimensionality, if you take two random vectors, they are almost orthogonal. And it's the orthogonality vectors that gives you.
So here's an example of how you would build up a scene vector then from this kind of a scene here, right? So right now we're currently glimpsing at the 6 in the center. So we would take the vector, the high dimensional vector we get for that 6 and what we're currently looking at, and bind it together by element-wise multiplication with the vector for where we currently are. So this is R is our current position, so we bind those two together, and we go into the next location and bind the representation of 5 together with where we're looking there, and then add that to the representation of 4 bound together with where we're looking there, right? Okay, we just add these all together, and now M contains a holistic representation of everything in the scene. Yeah.
So by the way, I don't know if you were Russ's talk, Russ Sullivan.
Yes, yes.
He was using this.
Oh, okay, I was going to mention that because there were a lot of talks here at the workshop, Russ is included, where they're sort of saying, oh, multiplicative things seem to help, right? And empirically, right? He sort of came across this, and so I think what's exciting to me here is that maybe this is a sort of gives us a theoretical way of understanding why. Yeah.
I mean, I thought about it and I'd come with the same explanation that you're telling from the thesis, and I went and told them that.
Okay. Oh, cool. Okay. Beautiful. Yeah. No, I think it's really neat. I think there's a lot of things converging here. That I think one of the lessons we're learning is that multiplying, representations together was useful. And here's that's right.
How do you know that to get the green box around a 6 or a 5? I mean,
okay, I'm going to talk about that in a second. But like in the draw network, it learns to do that, right? It learns how to move around. But here, right now, for the purposes of this illustration, we're just kind of randomly looking around the scene, just for this example. And then
you might not hit a 5. You might hit like half of a 5.
You might hit half a 5, and that's okay. It's going to still work. Okay. Yeah. But just right now, this is the toy example. Just to start out easy, right? And so here's an example of how you could build up this kind of scene representation. Once we have this now, we can start querying it in ways that we never trained on, right? We're not training whatsoever, actually. Yeah.
Is there any principal way to set the dimension of the representation space, or do you just do that empirically, just to see what works?
I think it goes back to what Penty said. As long as you're sort of somewhere between 1,000 to 10,000 is the sweet spot. So what you want is I'm about to show in a second. What you want is the property of high dimensional spaces where you can fit many orthogonal vectors.
Presumably it's some number that will trick that. That's my question.
So I think what Penty's found empirically is like below 1,000, it starts to sort of fall apart. Okay, you start to lose. Above 10,000 is sort of like diminishing returns.
And non-linearly orthogonal vectors grow exponentially with the dimensionality.
Yeah. Right. So I think below, so we want to be sort of above 1,000 kind of, but yeah.
Just a quick question. It seems that at least one of these must be very distributed, cannot be sparse, right?
Yes. That's right. These are entirely distributed representations. So like the representation of position, even though there's only 2 numbers in there, x and y, we're representing with 1,000 numbers, right? And that's good because it's going to allow us to do this kind of manipulation.
A lot of neurons have fire, for example.
That's right. It's going to be a very dense distributed representation. That's right. And OK, so here's an example of we can now ask it a query like, OK, fine. I'm almost done. So we can ask a query like, where is the 5 in the scene? Or what object is in the center? So how would you ask it, where is the 5? Well, that's very simply done in this framework by simply taking the memory vector and binding it with the representation, the conjugate, in case it's opposite of inverse of 5. Okay, so we bind it with this representation of 5. And what that does, we take that v5 star and bind it with this whole thing. What's inside of M, what's going to happen is that this will cancel, right? Because this is going to be orthogonal to that. This is going to be orthogonal to that. And so those go to 0. This matches that. So it comes out with identity of 1. And what comes out is the position of that object in the scene. If we want to know what object's in the center, then we take the same memory vector and we bind it together with a representation of center in the scene. And what that's going to do is, in the same representation of M here, it's going to match this one here. So R, at the first fixation, we were in the center. So I'll match R. It won't match the other two. So those other two go to 0, and it's going to return V6. Okay? So this is the kind of properties we're kind of looking for in this representation. I should have some kind of vector which represents what's in there holistically, but now is easily queryable, right? I can sort of pull out the contents of what's there in the scene. Okay, so I'm running out of time, so I have to go through this really quickly. This is just an example of building up a scene representation with a number of digits here, and this is just showing kind of an internal visualization of this internal representation M memory being built up. Each of these things, blue boxes here, shows you a spatial map of where the system thinks that particular digit is in the scene. This is the map of where the system thinks a 3 is in the scene. This is where it thinks a 7 is, and this is what's assigned to background. And in this case, Eric is using basically uncertainty to drive where to look next. So initially, the system is highly uncertain about everything in the scene, and so it just picks a random location to move to. But as it starts to build up a scene representation, it has high certainty at some locations and low certainty in others. And it chooses to look at places where it has the where it has the most uncertainty, right? Because that's where it's going to gain the most information. So that's what's basically driving it where to look in the scene. And as it does so, it sort of moves around the scene. It gradually builds up a representation of these different digits being present, and also where the background is in the scene, and it's pretty much and basically increases its certainty over time about that stuff. Okay, and then just the final example here, we can take a scene like this and ask it now a more complicated question like what is below a 2 and to the left of a 1. And basically, there's kind of an algebra of how you would take this question, parse it, and reframe that in terms of these hypervectors. And that's just shown here. First, you would say, well, what's below a 2? That's the way you would ask that question. And that's where it is. This is the way you would ask the question, what's to the left of a 1? And then you take the intersection of those two. That's this and here, right? And the and there is the intersection of those two. And once you query it with that location, it's going to return to you the answer 7. Okay, so what I've hoped to convey here is that vision in biological systems really works and is entirely defined in this active setting. Okay, there's no such thing to the system as a static image. We know that these movements help you even when you're trying to hold the eye still. They help you build up a better image of the scene, and this foveated sampling strategy that we see in many animals could be best explained in terms of this connection to how it's used in an active vision context to do search in the world. And finally, how you take all these sort of snapshots you get of the world and build up kind of internal scene representation. We think that binding is really an important part of that. The ability to take these bindings and combine them over time to build up some kind of a short-term memory of the scene to understand what's there and to do action in the world is really a necessary component. Okay, so I think that's it. Happy to take questions.
So there's like if I look at it like a grid, and how am I trying to phrase this? There's like an optical illusion where you look at a grid and you see these black dots which aren't actually there. Yes. I don't know if that's like in the brain or if that's part of how the eye hardware works, but can you sort of account for these optical illusions in your computational model?
That particular one, probably not. But you know, I would say all these illusions are in your brain. You see with your brain, not with your eye. And so that one particular one, it probably has something to do with people explain it in terms of center surround receptive fields. So maybe maybe you can explain it in terms of the retina in that particular case. But most of these illusions are sort of due to some kind of high-level filling in phenomenon. The brain is trying to impose its structure on the data that comes in. But that particular one, no, that doesn't address.
Is there any sense that there is similar structures in other sensory inputs, audio, tactile, that you have these kind of center focuses and kind of
Yeah. In the auditory systems of many mammals, they're sort of they put their sensors where the action is. So for us, it's between 20 and 20 kilohertz. For cats and dogs, it's much higher. For bats who use echolocation, their pings are sent out at about 60 kilohertz. And indeed, if you look at their auditory cortex, there's like a sort of fovea in the way around the auditory representation around 60 kilohertz. It's greatly magnified. Like all of its neural resources are devoted to 60 kilohertz, and then everything around that kind of diminishes and falls off. So, and then even in electric fish, use this electric field that they send out, and they have kind of like a foveated region of their electric sense organ where they're packing with very high density, where their nose is, where they kind of move it along the ground, and they're kind of looking for things, but there's a kind of fovea in the electric fish organ as well. So yeah, you see it like in all different, in the fingertips here, your sampling density is very large, very small. So you have very high resolution in your fingertips. That's why we do Braille there. On your back here, it's horrible. So it's like the periphery in vision. So, yeah.
All right, well, let's thank her.